Welcome to the Sand Science Experience podcast, where we explore the latest and most exciting papers and preprints in science. If you're finding our deep dives valuable, please remember to like this video and subscribe to our channel. Your support really helps the Sand Science Experience continue bringing you the most exciting science. Today, we're embarking on a journey into the very essence of time's impact on our biology. We're tackling a question that truly gets to the heart of what it means to live, adapt, and ultimately to age. How does the relentless march of time affect our bodies at their most granular, fundamental level, the single cell? It's a quest for the cellular blueprint of aging, seeking to uncover whether there are universal rules written in our cells regardless of species. To unlock these secrets, we're diving deep into 3 groundbreaking papers. These studies have harnessed the revolutionary power of single-cell RNA sequencing, or cRNA-seq, to construct intricate cellular atlases of aging in 3 classic, yet remarkably diverse, model organisms. We've got the humble worm, C. elegans, the ubiquitous fruit fly, Drosophila melanogaster, and, of course, our close mammalian relative, the mouse, Mus musculus. It's a scientific adventure into the cellular landscape of time itself, unveiling the hidden dynamics of decline and resilience.
It really is.
Yeah.
And for those of you maybe less familiar, single-cell RNA sequencing, a cRNA-seq, it's truly a game changer. Historically, researchers would take a tissue, say, a piece of liver, grind it all up, and then sequence all the RNA together.
Right, the bulk approach.
Exactly. It's like trying to understand a city's economy by just looking at the average income. You get some information, but you miss all the diversity, right? You don't know if it's tech booming or manufacturing struggling.
Yeah, you lose the granularity.
Precisely. cRNA-seq lets us peer into individual cells within that tissue. It's like interviewing every single business owner. And this resolution is absolutely crucial for understanding aging because, well, we now know different cells, even in the same tissue, age differently.
At different rates, with different signatures.
Exactly. It's not a uniform decline. It's much more complex, like a symphony of cellular change, really.
That distinction is paramount. And what's particularly vital for our deep dive today is our focus. We are exclusively looking at the findings from wild-type animals in these studies. So no mutations, no treatments, just the natural aging process.
Right. Observing what happens when time simply marches on. This gives us that essential baseline, that map of inherent changes.
It's the foundation we need before we can even think about interventions.
So our mission for you today is pretty clear. We're going to systematically navigate through each of these 3 landmark papers. First, we'll spotlight what each study discovered in its organism: mice, worms, flies. What are the key cellular shifts? Which tissues are most affected?
Okay.
Then we'll pull back and look for the shared patterns. Are there universal mechanisms emerging across these species?
The conserved features.
Exactly. And finally, we'll pinpoint the unique insights each model provides. What does the worm tell us that the mouse doesn't, and vice versa?
Sounds like a plan. Discerning the common rhythms and the unique solos in the aging symphony?
Couldn't said it better myself.
Okay, let's kick things off with Musculus, the mouse, a real workhorse in aging research. We're looking at the Tabula muracenis, the Mouse Aging Cell Atlas. This sounds incredibly ambitious.
Oh, it is. Audacious is a good word. Think of it like building a Google Maps of aging for an entire mammal, cell by single cell. They set out to generate a single-cell transcriptomic atlas spanning almost the entire natural lifespan of mice.
Giving us this super high-resolution view of how aging unfolds in a mammal.
Exactly. And the scale. It's staggering. They analyzed data from 23 different tissues and organs.
23, wow. So everything from brain, heart, liver.
Kidney, spleen, even tongue and bladder.
Yeah.
All in, over 350,000 individual cells.
Okay.
Sourced from both male and female mice across 6 age groups, ranging from 1 month old, like early childhood, all the way up to 30 months.
Which is like a human centenarian,
Pretty
basically.
much, yeah. A really comprehensive age spectrum. And to do this, they used a smart dual method approach, a droplet-based method for analyzing huge numbers of cells quickly.
High throughput.
Right. And then fluorescence-activated cell sorting, FACS, for targeting smaller populations, but getting higher sensitivity and full-length transcript info.
So quantity and quality.
You got it. And crucially, this wasn't just automated processing. They had rigorous manual curation. Experts went in and meticulously annotated cell types.
That human touch is so important for accuracy.
Absolutely. It makes this atlas incredibly robust.
Yeah.
One of the largest manually curated single-cell resources out there.
That's impressive. And I think you mentioned something really unique about how they collected the samples.
Yes, this is a key strength. They analyzed multiple organs from the very same mouse.
Ah, okay. So that controls for individual differences, like environment or subtle genetic variations.
Exactly. But taking a full suite of organs from one individual at each age, they controlled for all those confounding factors.
It's like getting a complete checkup on one person over decades, but at the cellular level.
Precisely. It gives this incredibly comprehensive, internally consistent view of aging across the whole system.
Which lets you really trust the comparisons between organs.
Right. And with this single-cell resolution, they could then assess how the well-known hallmarks of aging manifest in specific cell types. Let's start with cellular senescence, where cells stop dividing but hang around, causing trouble.
Yeah, the zombie cells.
Kind of. They found the proportion of cells expressing the key senescence marker, CDKN2A or P16, more than doubled in older mice.
Doubled. That's significant.
Very. And it was consistent across both their sequencing methods. At the same time, genes in the sirtuin family, SIRT3, SIRT4, SIRT5.
The ones known for promoting cellular health and delaying senescence.
Those are the ones. Their expression significantly decreased with age. So you see this inverse relationship, this molecular shift towards senescence.
A decline in resilience paving the way for those senescent cells.
Okay. What about genomic instability?
DNA damage.
Another big one. They found clear evidence of an age-related increase in somatic mutations. DNA changes after conception across all 23 organs.
All of them. Wow.
But here's the specific insight. The tongue and the bladder were the most affected.
The tongue and bladder. That's unexpected. Why them?
Well, think about it. Constant mechanical stress, exposure to substances or waste, maybe higher mutational pressure, or perhaps less efficient DNA repair as they age.
Interesting point.
And they were careful to validate this wasn't just a sequencing artifact, it's a real biological finding.
Okay. And the third area, the immune system. Always a big player in aging.
Absolutely. They saw profound shifts here. They looked at B-cell and T-cell clonality, basically how many specific immune cell lineages expand. And they found clonality increased significantly from 3 months to 24 months.
Meaning the immune system becomes less diverse.
Exactly. Less diverse, less flexible, less able to respond effectively to new threats.
Which lines up perfectly with what we see in older humans more vulnerable to infections, less response to vaccines.
Precisely. It's a clear sign of immunosenescence, the aging of the immune system itself, captured at the cellular level.
Okay, let's unpack this further. You mentioned specific tissues showed really pronounced changes. Let's dive into some of those. The bladder, for instance.
Right, the bladder was a great example of major shifts in cell composition. The mesenchymal compartment, the connective tissue providing structure, it decreased threefold with age.
Threefold decrease. Wow.
While the urothelial compartment, the inner lining, increased by a similar amount. This matches known age-related bladder changes, like stiffness.
And the gene expression reflected this.
Beautifully. Connective tissue genes like collagens, COL1A1, COL1A2, COL3A1 went down. Epithelial genes like keratins, CART15, CART818 went up.
The bladder's architecture is literally reshuffling.
It is. Plus, they saw fewer endothelial cells, suggesting less blood supply.
Which can't be good for tissue health.
Definitely not.
Yeah.
And more leukocytes, more immune cells hanging around, signaling an inflammatory environment.
Inflammation again.
Yep. These leukocytes showed pro-inflammatory gene signatures, too. It's a clear picture of structural and immunological decline.
Paints a concerning picture. How about the kidneys? Another organ prone to age-related issues.
Kidneys, too. They saw an age-related decrease in 2 key cell types in the glomerulus, the filter unit, mesangial cells and capillary endothelial cells.
So the core filtration machinery is declining.
Seems like it. This, plus lower expression of genes like AGE and ADP1A1, the sodium-potassium pump, strongly suggests impaired glomerular filtration rate, a hallmark of kidney aging.
Okay.
But here's a subtle twist from the single-cell data. While overall AP1A1 expression decreased, its expression actually increased locally within those declining cell types.
Like the remaining cells are trying to work harder. A compensatory effect.
That's the hypothesis. Like fewer workers try to pick up the slack. Desperate measures, perhaps.
Fascinating detail.
And they also saw reduced UMOD expression uromodulin, another sign of impaired kidney function. It's a comprehensive decline.
And were there similar patterns elsewhere? Specific cell declines in other organs?
Yes, several examples. In the spleen and the mammary gland, they saw a decline in T-cell populations.
T cells again, linked to higher risk of infection and cancer.
Exactly, a consistent finding. In the mammary gland, they also saw upregulation of AP1 transcription factors, consistent with age-related involution or shrinking. The liver also showed changes. Fewer hepatocytes, the main liver cells, backed up by lower albumin expression.
Okay, reduced liver function.
And notably, an increased immune signature throughout the liver. The number of Kupffer cells, the liver macrophages, didn't change,
So
but
let me guess, they became more inflammatory.
you got it. More Kupffer cells expressing the pro-inflammatory marker IL-1B. So not more immune cells, but a shift in their state.
A shift towards inflammation. That theme keeps popping up.
It really does. Changes in other liver cells, LSECs, also pointed towards altered immune responses.
Okay, now here's where I think it gets really interesting, especially thinking about brain health. What about microglia, the brain's immune cells?
Ah, yes, this was a truly standout finding. Huge implications. They saw significant, very specific changes in brain microglia. Young microglia occupied certain gene expression clusters, a healthy state. But old microglia, they were in distinctly different clusters.
A different molecular identity entirely.
Completely. Trajectory analysis even suggested they go through an intermediate state. And critically, these old microglia upregulated MHC class I genes.
Immune activation signals.
Right. And genes linked to degenerative disease, like FTEC1, plus interferon response genes, all point to an expansion of a pro-inflammatory microglial subset.
Pro-inflammatory microglia building up in the aging brain.
And here's the kicker. The gene signature of these old microglia strikingly resembles the Alzheimer's disease-specific microglial signature.
Wow, so natural aging in the mouse brain makes microglia look like those in Alzheimer's.
Exactly. It provides this direct cellular link. Aging itself might be priming the brain's immune system for disease.
That's quite profound. A potential mechanism for age-related neurodegeneration risk.
It really is.
Yeah.
And this pro-inflammatory shift wasn't just in the brain. Kidney macrophages showed a similar transition. They shifted from an M2 signature anti-inflammatory repair in young mice to an M1 pro-inflammatory state in older mice.
M1 is the attack mode.
Basically, yes. So this systemic bias towards inflammation, driven by specific immune cell, changes across different organs. It suggests this broad, low-grade inflammation, inflammaging, is a key driver of age-related issues.
So bring it back to the listener. What's the big takeaway from the mouse atlas? It's not just that organs get old. It's about which cells change, how they change, their proportions shifting.
Precisely. That microglia finding, for example, directly links normal aging wear and tear to microglia looking like those in Alzheimer's. It's a foundational connection.
And this detailed cellular map is crucial for finding specific targets.
Absolutely. It helps us identify the cellular weak points, the places where interventions might actually promote healthy aging or prevent disease at its earliest stages. It's a roadmap.
Okay, from the complexity of the mouse, let's shift gears to something much smaller but just as revealing. The worm, C. elegans. We're looking at the Cell Atlas of Worm Aging, the CIWA study. They used a slightly different method here.
That's right. Primarily single-nucleus RNA sequencing, snRNA-seq. It's apparently less biased for certain worm tissues that are tricky to dissociate, like muscle or the hypodermis, their outer layer.
Okay, it gets a truer picture for those tough cells, and they looked across the worm lifespan, too.
Yep, 4 adult ages. Day 1, young, Day 6, early adult, Day 12, midlife, and Day 14, late life. Covers the whole physiological journey. And from thousands of nuclei, they identified 15 major cell classes: neurons, glia, hypodermis, intestine, muscle, pharynx, germline. Really broad coverage.
Gives a holistic view of the whole tiny organism.
It does. And they introduce this insightful concept, transcriptome drift.
Transcriptome drift. Sounds like how much the gene expression profile changes or drifts away from the young state.
Exactly that. It quantifies how much a cell type's molecular score changes with age. A bigger drift means more molecular change, more sensitivity at aging.
Okay, so which tissues showed the most drift, especially early on?
This was really interesting. A hypodermis in neurons showed significant drift as early as Day 6.
K6. But the worms are still mostly healthy then, right? Almost 100% survival?
Correct. These big molecular changes were happening very early, long before widespread decline. Suggests these tissues are more sensitive early on.
Maybe early indicators or even drivers of the aging process, like the first cracks appearing?
That's a good analogy, but here's where it gets fascinating regarding later stages and survival. The drift in neurons, which was high early on, actually decreased on Day 12 and 14.
Decreased? How does that happen? Do they get younger?
Not quite. The likely explanation is, well, selective survival. Individuals whose neurons drifted too much might have simply died off.
Survival bias. The ones left are the more resilient ones, skewing the average.
Precisely. So the remaining population looks like it has less severe neuronal aging because the worst affected are gone.
That makes sense. A sobering reminder when studying populations. Did other tissues show this?
Not like the neurons. The hypodermis drift just kept increasing until day 12.
Yeah.
Muscle drift picked up later, around day 12. Pharynx and vulvatorus only drifted much later, day 14.
So really different aging speeds. And what about the intestine?
That was the big surprise. The intestine showed no obvious drifts even on day 14. Remarkable transcriptome robustness.
Wow. So the gut stays molecularly stable right to the end. That's amazing resilience. Why?
Great question. Maybe intrinsic resistance, maybe super efficient repair mechanisms. It's a key area for future research.
It really highlights this multi-speed aging within one organism. Some systems falter early, others are incredibly robust.
Definitely. Now, shifting to the germline, the reproductive tissue, studying its aging is tricky because it's constantly proliferating and differentiating.
Right, not like most body cells that just sit there.
So they developed these germ cell trajectory maps to track cells from stem cell state through meiosis to mature oocytes and see how that pathway changes with age. And they found clear compositional changes. Fewer germ nuclei in the transition zone of meiosis region between day 6 and day 12.
Critical stages for making reproductive cells. So a decline there impacts fertility.
Directly. And molecularly, they saw interesting patterns. Ribosomal protein genes crucial for making proteins.
Yeah.
Their expression, normally restricted to the stem cell end, actually expanded towards the other end with age. This might reflect problems with differentiation, like the cells aren't maturing properly. Plus, genes for lysosomes and mitochondrial cellular recycling energy showed decreased expression in the distal germline. Suggests declined organelle production and maintenance.
The cell's internal machinery is struggling.
Seems like it. And at the other end, where oocytes mature, genes for DNA synthesis, chromosome assembly, protein folding, mRNA processing all decreased with age.
Which likely explains the lower quality of aged oocytes.
Exactly. It connects the molecular changes directly to the functional decline in reproduction.
The study also developed aging clocks based on this data, right, using machine learning.
They did. Using lasso regression, they built models that could predict the worm's chronological age very accurately just from tissue-specific gene expression, R-squared values over 0.90.
That's incredibly accurate. Shows the power of these atlases for quantifying aging.
It does, and what's fascinating about the aging clock genes, the ones the model used for prediction, is that they were mostly tissue-specific.
Very little overlap between tissues.
Extremely little. Only 2 genes were shared aging clock genes across all tissues. A ribosomal RNA, MTCE.7, and a non-coding RNA, CO5D92005.
Wow. Really emphasizes the heterogeneity of aging at the molecular level.
Totally. And the functions of these clock genes made sense. Neuron clocks involved ribosome and autophagy pathways, muscle clocks in motor protein pathways. Metabolism was enriched almost everywhere, but with different genes involved.
So different tissues age differently with unique molecular signatures, no single master clock program.
That's the clear message. And they looked at another layer, too. Alternative polyadenylation, or APA.
APA. That affects mRNA stability and translation, right? How the genetic message is processed and used.
Precisely. It allows one gene to make multiple mRNA variants by choosing different ending points.
Fine-tuning gene expression.
Exactly. They found hundreds of genes showed age-related APA changes, and for over 70% of these, there was an increased use of distal APA sites as the worms got older.
What does that mean in aging cells?
Well, in young cells, distal APA is often linked to differentiation. But in these older, mostly non-dividing cells, this widespread shift suggests a global regulation of APA by aging. It implies fundamental shifts in mRNA stability and how efficiently proteins are made from those messages. It's not just which genes are on, but how they're processed.
So taking it all together for the listener, the worm study shows aging is incredibly complex, even in a simple organism. Different tissues aging at different speeds, specific organelles declining, even fundamental changes in how genetic info is processed via APA.
Right. It reveals this multifaceted, highly tuned process and really hammers home that aging isn't simple, uniform decay. It's written in these surprising molecular details.
All right, from the worm, let's move to our final model, the fruit fly, Drosophila. Another powerhouse in genetics research. This is the Aging Fly Cell Atlas, AFCA. And this one sounds massive in scope, too.
Oh, absolutely monumental. A single nucleus transcriptomic map of the whole aging fruit fly. They characterize 163 distinct cell types, 91 from the head, 72 from the body, analyzing over 868,000 nuclei.
Whoa, nearly 1 million nuclei.
Yeah. across 4, ages 5, 30, 50, and 70 days, covering young adult to very old. And they looked at males and females separately.
Which lets you look for sex differences in aging. Very cool.
It does. And a key baseline finding was that overall, the organism largely maintains its cell types during aging.
Okay, so it's not like whole cell types are just disappearing en masse.
Generally not. It implies aging is more about functional changes within existing cells or shifts in their proportions rather than massive cell loss.
Makes sense. But they did see changes in the proportions, the cellular composition.
Profound changes, yes. And here's a really novel finding on the increase side. A significant increase in fat body nuclei as flies aged.
Fat body. That's like the fly's liver and adipose tissue combined, right?
Exactly. Roles in metabolism, energy storage. And these cells are usually post-mitotic. They don't divide.
So how did the nuclei increase?
Through this fascinating process called nuclear cleavage without cytokinesis amitosis. The nucleus divides, but the cell itself doesn't.
So you get multinucleated cells, more nuclei packed into one cell.
Yep. A really unexpected cellular adaptation or response to aging stress, maybe to boost metabolic capacity. We don't fully know why yet.
That is fascinating. A completely new way a cell might cope with aging.
It really is. They also saw increases in some gut cells, male accessory gland cells, intestinal stem cells, and salivary gland cells.
Okay. What about decreases?
A big one was a decline in muscle cells, especially the indirect flight muscles.
Flight muscles. So that links to loss of movement like sarcopenia in mammals.
Exactly. Consistent with sarcopenia. And they found increased Caspase-3, an apoptosis marker, in those old muscles. So more programmed cell death.
That makes sense.
Also, a decrease in germline cells, contributing to lower fertility, as expected. And interestingly, like the mouse, head cells, mostly neurons, showed minimal composition changes, relatively stable.
Okay, but that fat body finding, nuclear division without cell division, that's the real headline grabber there. It just changes how you think cells might respond.
It really does. A prime example of how these Alices uncover novel biology.
So beyond cell numbers, what about gene expression within the cells? Were there hotspots of change?
Yes. The fat body and outer photoreceptor cells in the head had the highest number of differentially expressed genes, DEGs.
So those cells are undergoing the most significant transcriptional remodeling with age.
Seems that way. And it confirms again, different cell types age at different rates with distinct patterns. A lot of changes happen relatively early, between 5 and 30 days.
Mid-adulthood is when a lot of the action happens molecularly.
Appears so. Some cells, like male accessory gland, changed fastest early on. Others, like intestinal stem cells, showed changes later, different trajectories.
And they looked at sex differences too.
Yep. Female marker genes, related to egg production, decreased significantly in most female cells. Male markers were more stable, and some genes just showed different age trends between sexes. Clear sexual dimorphism.
Okay. Was there anything universally changing across all cell types?
Yes, and this is huge. A globally shared downregulation of genes related to cytoplasmic translation, protein synthesis.
Protein synthesis going down everywhere, across almost all cells.
Pretty much. Many ribosomal protein RP genes were decreased, consistent with a general reduction in the cell's ability to make proteins.
That sounds fundamental.
It is. And interestingly, while protein production went down, there wasn't a universally upregulated process across all cells, not just a reallocation of resources.
Got it. And tissue-specific changes, did they make sense?
They did. Fat body cells upregulated lipid and triglyceride metabolism genes. Makes sense. Muscles downregulated locomotor behavior and muscle contraction genes. Matches the functional decline.
And like the worm study, they built aging clocks for flies too.
Yep, highly accurate ones, predicting age from the transcriptomic data with R-squared values around 0.8.
Again, validating this approach for quantifying aging. Which cells were the best predictors?
Outer photoreceptor cells and enocytes, those liver-like cells, gave the highest prediction scores. Again, most aging clock genes were cell type specific.
Reinforcing that heterogeneity.
But here comes the really pivotal cross-species finding. Remember the ribosomal protein genes declining everywhere?
Yeah.
A remarkable 70 out of 94 fly ribosomal protein genes were identified as aging clock genes, showing age-related reduction.
Okay, so the protein machinery decline is key to predicting age in flies.
And here's the connection. They compared these to the mouse atlas. 31 of these fly ribosomal protein genes had direct one-to-one orthologs in mice.
That were also aging clock genes in mice.
Exactly. This is powerful evidence for an evolutionarily conserved mechanism of aging tied to the decline of protein synthesis machinery, shared across insects and mammals.
Wow. That's a major conserved theme emerging.
It really is. The study also looked at a couple more complex features. Changes in the number of expressed genes per cell,
Right. What did they find there?
a mixed bag. About 80% of cell types showed a decrease, as you might expect, but 20% showed an increase,
An increase, so some cells get more transcriptionally complex.
potentially. Het immune cells and certain fat cells showed the biggest increase. Enocytes and fat body cells showed the biggest decrease. Highly cell type specific responses.
And the other feature, cell identity decline. What's that?
It's a measure of how much a cell loses its original defining marker genes and gains new, unrelated ones, like it's forgetting its specialized role.
Getting less distinct, maybe more dysfunctional.
That's the idea. Indirect flight muscle, pericerebral fat mass, and some head epithelial cells showed a large decrease in this cell identity metric.
Okay, so when they put all these different aging features together, composition, DEGs, gene number, identity decline, could they rank cells by aging rate?
They could. And the results were surprising. The top 3 fastest aging cell types were all adipose cells: enocytes, fat body cells, and pericerebral adult fat mass.
Fat cells age the fastest overall. That's counterintuitive.
It is. In contrast, neurons and glia generally age slower based on these integrated metrics.
Challenges the idea that the brain is always the most vulnerable, maybe.
It certainly provides a new perspective. Suggests adipose tissue might be a key driver or hotspot of aging.
So for the listener, the Fly Atlas reinforces the complexity, shows this unexpected fast aging in fat cells, and provides that crucial cross-species link about declining protein synthesis.
Exactly. It gives this incredibly detailed map highlighting unexpected vulnerabilities in conserved mechanisms, really pushing our understanding forward. #tag cross-species comparison and synthesis.
Okay, let's try to pull this all together. We've gone through the mouse, the worm, the fly, amazing detail from each. What are the big overarching themes? The conserved features of aging that cut across these diverse species.
I think the most striking, the one that really jumps out, is the decline in protein synthesis machinery. We saw it clearly in flies with ribosomal protein genes as key aging clock genes and decreased translation overall.
Right, that direct link.
In mice, the sirtuin decrease points towards impaired cellular maintenance, which heavily involves protein quality control. In worms, ribosomal gene expression was altered, linked to differentiation issues. And that direct ortholog connection for RP genes between flies and mice as aging clocks, that's huge.
So a fundamental slowdown in the cell's ability to build and maintain itself seems universal.
It really looks that way. It's a core evolutionarily conserved mechanism, a slowdown in the cellular engine.
Okay, what else?
Tissue-specific aging rates and sensitivities. This came through loud and clear in all 3.
Yeah, definitely. Worm hypodermis and neurons early, intestine late. Fly fat cells fastest, neurons slower. Mouse bladder, kidney, specific immune cells.
Exactly. Aging isn't monolithic. Different parts decline at different speeds for different reasons. We have to look at specific cell types.
Agreed. What else was common?
Changes in cellular composition. The relative proportions of cell type shifted in all 3 models. Mesenchymal versus urothelial in mouse bladder, germ nuclei regions in worms, that multinucleation in fly fat bodies. The tissue landscape itself remodels with age.
Right, the cellular makeup changes.
And then there's immune system dysregulation and inflammation. Most obvious in the mouse with those pro-inflammatory microglia and macrophages resembling disease states.
The inflammaging theme.
Yes. While maybe less explicitly detailed in worms and flies, their gene expression changes often hinted at similar underlying processes. Suggest chronic, low-grade inflammation is a common thread.
Okay, and one final point.
Just the effectiveness of single-cell aging clocks. The ability to predict age accurately from this data was consistently shown across all 3. It validates this whole approach for quantitatively measuring aging.
Powerful tools for future research. So those are the common threads. But each model also gave us unique insights, right? Things we learned specifically from the mouse or the worm or the fly.
Absolutely. Each brings something unique to the table. The mouse, being a mammal, gave us fantastic detail on classical hallmarks like senescence and genomic instability at cell type resolution, that p16 increase, the tongue bladder mutations, and those specific immune shifts, especially the microglia looking like Alzheimer's microglia. That direct link between healthy aging and disease pathology is incredibly valuable from a mammalian model.
Right, the translational relevance there is high. What about the worm?
The worm offered exquisite detail on germline aging. Tracking that whole differentiation pathway and seeing how it falters was unique. And it uniquely highlighted the role of alternative polyadenylation, APA, shifts that fundamental change in mRNA processing as a global aging feature. Plus, that intriguing dynamic of transcriptome drift suggesting survival bias.
Yeah, the APA finding was really cool. And the fly.
The fly revealed that totally novel phenomenon of fat body multinucleation through amitosis, a new cellular coping mechanism perhaps. It also provided that comprehensive ranking of cell types by overall aging rate, pinpointing adipose cells as unexpectedly fast aging and neurons glia as slower. And the concept of quantifying cell identity decline was also a unique contribution.
So synthesizing all of this, these atlases move us way beyond just saying an organ is old.
Totally. We're now understanding the precise cellular and molecular changes. It gives us much richer, more nuanced picture. We see both the universal mechanisms like declining protein synthesis and these remarkable tissue and species-specific details.
It's like moving from a blurry map to a high-definition satellite view with GPS coordinates for aging, showing exactly where the problems are.
That's a great way to put it. This kind of detailed map is essential. It pinpoints the vulnerable cells and processes.
Which is the crucial first step towards figuring out how to intervene effectively and promote healthier, longer lives. Hashtag tag 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 outro. Well, that was quite a journey through the cellular landscape of aging. From worms to flies to mice, all viewed through the incredible lens of single-cell sequencing. We've seen how different cells age at their own pace, uncovered conserved signatures like that protein synthesis decline, and found surprising insights, fast-aging fat cells, microglia mimicking disease. Just amazing detail. Thank you so much for joining us on this deep dive. If you enjoyed exploring the science with us, please do hit that like button, share this with anyone who might find it interesting, and subscribe to the Send Science Experience. Your support really does help us keep bringing these cutting-edge explorations to you.
And it leaves you thinking. As we keep refining these cellular maps of aging, getting ever higher resolution, how will our fundamental understanding of longevity and age-related disease transform? What new pathways for intervention, things we can barely conceive of now, will open up?
A fascinating thought to end on. Until next time, keep exploring, keep questioning, and keep deep diving into the wonders of science with the Science Experience.